Great. Well, thank you so much, everyone, for coming out here today. It's really a pleasure to tell you about the research I'm doing in the lab. A lot of what I'll talk about is cancer biology, cancer therapeutic research. But I thought I'd start with, for me, where it really all begins, really why I have a career in biology in the first place. And I thought I'd start with an area of biology that I think everyone would claim to be an expert in, which is sex. So I think what has impressed me when I first learned about biology when I was 12 years old was how life has been perpetuating for billions of years on this planet through cycles of reproduction. And the combination of the DNA from both parents are transmitted to a single cell. And that one cell is able to, guided by the instructions in their DNA, give rise to the body plan for life on Earth. And of course, this includes humans. But of course, all life on Earth shares this same genetically encoded biology. And underlying this, of course, is DNA, a relatively simple biopolymer that includes 4 different letters, information encoding letters to its makeup, but are arranged in including billions of letters in length. And encoded in this sequence is body plan biology information. Now, and I think what is, of course, extremely impressive about this process is how one single cell containing this genetic information can multiply itself to give rise to an embryo, which can later become a human being. During this process, this genetic information, effectively a genetic hard drive, is copied every time a cell will multiply. However, during this process, there are always genetic mistakes that occur, and I'll come back to that. So this multiplicative process for cells is profound, and it occurs very quickly. What is also extremely impressive about this process is how one single cell, as it's multiplying, can take on a specific identity, which ultimately gives rise to the unique functions of each organ in the body. And this is really the area of biology that my lab focuses on, which I'll be referring to as epigenetics. And what this means is that the same DNA genetic information transmitted from mom and dad is contained within every single cell in the body in each of these organs. But the way in which this DNA is read and executed upon is highly organ-specific and cell-type specific. The way you can think about this is that DNA is the hard drive of genetic information passed down for millions and billions of years. But that hard drive is only accessed in the right cell at the right time. And how that works is the subject of the research of my laboratory. And really, a key way to think about this is that all these genes exist encoded in DNA. Certain genes in a cell, certain genes in the body are only turned on in the right cell type. So brain genes are only turned on in the brain, heart genes in the heart, and so on and so forth. And so the core biology of my lab is how molecules act inside of cells to turn genes on and off at the right time and place. Now, this is sort of the problem that a cell encounters, is that there's this genetic code, 6 billion letters in length, with, again, simple 4 different letters arranged in a sequence. And this is the body plan for human life. And the challenge that a cell has is that it has to find order and structure in this rather repetitive, simplistic sequence, which includes the identity of those genes I was telling you about that get turned on at the right time and place. The cells in the blood have to identify the right genes and turn them on out of this simple code. And how this works is that inside of cells, there are biomolecules that are able to decode and read the DNA sequence. One of these is depicted here. So this is a molecule so small you can only see it with the most sophisticated electron microscopes, sitting on the double helix of DNA, and it's reading that DNA sequence and essentially decoding the DNA and telling the cell that this is a liver gene or this is a heart gene. And so what I have devoted most of my lab's research to, again, for me going back to when I was even an undergrad researcher, is how this works. How does a cell read out the DNA and execute on the generation of specific cells and organs? This process is what's often referred to as epigenetics. It's the turning on of genes in the right time and place. And the best way I could describe what we do all day is depicted here. So we don't make movies of molecules for a living. This is an artist's rendition of what molecular biology looks like. We use technology in the laboratory to see what molecules do. Our cells are filled with thousands of molecular machines, some of which are depicted here, accurately depicting how we know they operate in cells. But inside of our cells is this elaborate, dynamic set of machines that coordinate and orchestrate and animate life. And what we do is try to see what's happening, to try to reverse engineer and understand how these molecules work. And I think the importance of this research, which is shared by me with many of my lab colleagues at Cold Spring Harbor, we know that an error in one of these molecular machines inside of a cell is sufficient to generate a human disease. And so that knowledge has informed us over the last 50 years. We know the molecular defects, how the car becomes broken to cause almost every human disease. And I think equally inspiring is that we've been able to generate medicines that can change and fix these molecular machines when they're broken in a way to rectify and correct many human diseases, with many more yet to be solved. Okay, so how does this relate to cancer? So as I've told you, this elaborate expansion of one cell to billions involves copying DNA, but that process is not completely accurate. Every time this happens, that DNA that you inherited from mom and dad receives an error or a mistake. And rarely, a cell can have a catastrophic mistake, where a terrible combination of genetic defects occur. And that is what sets in motion a single cell multiplying out of control. And this is what drives the pathobiology of cancer. However, the key aspect of cancer biology I want to tell you about from our work is how each cancer not only originates in a single organ. You're aware that every cancer is classified by the organ they come from. But the cell, the identity, the epigenetic on-off state of genes in that cell dictates the behavior, the biology, and the way you can treat each form of cancer. Each type of cell has an epigenetic memory of its identity, and the tumors that emerge from those cells retain that identity. And you can take advantage of this identity in interesting ways I'll tell you about. And this identity is it can be stable, but it can also be plastic. And really the key thing we can talk more about later is that the epigenetic memory, the identity that a tumor has profound implications for cancer therapy. If any of you know of someone that's been treated for breast cancer or prostate cancer, frontline therapy for those two very common malignancies are drugs that manipulate the epigenetic state of that tumor. So you're all aware that chemotherapy, radiation therapy, surgery are still how most human cancers are treated. Those types of therapies, which are common but limited, do not take advantage of this molecular understanding. Those are therapies that truly have no molecular intelligent design underlying how they work. But what many of us are pursuing and are most excited about is how our knowledge of epigenetics on a molecular level can allow for more efficacious, safer therapies that don't have the catastrophic side effects of these more surgery, of radiation, of chemotherapy. And so as I said, frontline therapy for several common cancers is epigenetics exploiting. Hormone therapies for prostate and breast cancer, immunotherapies, taking one's immune system, educating it to eradicate one's tumor, is also exploiting an epigenetic memory that a cancer cell will have. And so many of the cutting-edge therapies that many of us are working towards and are developing and already in the clinic, this next generation that we seek to replace standard chemotherapy, is exploiting this type of molecular knowledge. We'll talk more about that. So one topic that we've been infatuated with, obsessed with, and kind of frightened by is shown here. So one cancer we put a lot of effort into is called pancreatic cancer. It's a cancer of the pancreas. As you would expect, the tumor, the cell that acquired that genetic catastrophe is a cell of the pancreas. And so based on everything I just told you, the pancreas tumor should have an epigenetic memory of being a pancreas cell. The pancreas is an organ that produces digestive fluid. One observation that's been made by physicians, pathologists, is that when you look inside of these tumors with a microscope, as shown here, one will find kind of the epigenetic memory I just told you about, that the tumor will have the appearance of pancreas tissue, which makes sense. It's a pancreas cancer. It should look like a pancreas cell. But what has been found is that one can find skin cells growing as a part of this cancer inside of the pancreas. Skin cells, as you know, do not exist in the pancreas normally. But the cancer is changing its identity, changing its epigenetic state, the pattern of genes being off and on, to become like skin. And what has been learned is that when a tumor starts to acquire a skin appearance, these patients die much more rapidly than patients that die with the tumors that lack skin. So it correlates with bad prognosis. These patients are much more metastatic. They do not respond to chemotherapy as well as tumors that lack skin. And so one big focus of our research has been to understand, is this just a correlation or is this causation? So does skin, when this happens, does this actually cause the tumor to be more aggressive? And so over the last 8 years, we've addressed this question and shown that in the laboratory, when we make a pancreatic cancer form skin type cells, this makes the tumor more metastatic, more chemo-resistant, and more aggressive, establishing that this change in cell identity is making the disease more aggressive. We've been revealing the molecules that drive this, and we've been working towards therapies that might eliminate the skin biology as a way to suppress the pathogenesis of this disease. And so I just use this as an example where cancer cells don't read the textbook about epigenetics and how cell identity is specified. Cell identity is malleable, and cancer cells can kind of change their biology to gain more aggressive attributes. And so that's an example where cell identity changes in a way to make cancers worse. But can you exploit this to make cancers better? And so a topic my lab has worked on for almost since I started my lab at Cold Spring Harbor 15 years ago, is a blood cancer called acute myeloid leukemia. This is a cancer of white blood cells that start and take over the bone marrow, a highly aggressive disease, one of the most common cancers of childhood. Childhood cancers are a large focus of our research. These are what leukemia cells look like. They are kind of immature cells. These are cells that should have become white blood cells that fight infections and eliminate bacteria. Unlike normal white blood cells, which look like this, they're arrested. They cannot complete their transition to become a normal white blood cell. They are immortal. They can grow forever. That's why they take over and crowd out the bone marrow. They divide rapidly, and the immune system cannot detect these as being a cell that doesn't belong, in part because these cells have eliminated the immune system by crowding out your normal blood production. The idea that our lab and many others have pursued is that maybe we can, instead of using chemotherapy to just poison all proliferating cells in the body, could we develop therapies that help these leukemia cells remember how to become a normal white blood cell again as an alternative to chemotherapy? And the idea is that normal white blood cells do not multiply, which would obviously be the opposite of the cancer cell. And these cells only live for a few days. And that part of that is because the immune system, once it's present, can eliminate them, get rid of them. And so this has been an idea to change cancer cells back to normal cells. We call this differentiation therapy. And going back several years ago, my lab discovered 2 epigenetic targets that allow for differentiation therapy in leukemia. We published these almost 10 years ago. It's kind of the launch discoveries of my lab. Both of these targets were, after we published them, were picked up by the pharmaceutical industry, who developed drugs that manipulate these molecular machines that we identified, which when targeted, cause this change to occur. These targets are called, these are their names. And what's exciting about this is drug companies brought these targets to the clinic and initiated phase 1 clinical trials, bringing these into patients. What's shown here is, to my knowledge, the very first leukemia patient treated with a drug that targets BRD4, one of these epigenetic machines that we identified as a target. This is a 74-year-old woman with acute myeloid leukemia who had failed 3 prior chemotherapy regimens, whose bone marrow at the time of this trial was 100% taken over by these immature leukemia cells that had shut off her own blood production. And what's shown here is the measurements of her leukemia cells in her bone marrow, and then in her peripheral blood, the emergence of normal-looking white blood cells on different days after treatment with this inhibitor, a pill that targets and reprograms the epigenetic state of the leukemia. And what you're seeing is a complete remission of her disease. And as we would expect, the transition of malignant leukemia cells to normal white blood cells, this time in a patient, and this has been seen in several patients on this trial. And so that was several years ago. Since that time, these drugs have been completed phase 2, phase 3 clinical trials as of last year, and are kind of in the on-deck circle for potential FDA approval for acute myeloid leukemia patients. And so what I want to emphasize, though, is the time. These were papers we published over 10 years ago. We don't even work on these targets anymore. We've been hunting for more targets in more cancers. It takes a long time for discoveries to happen in an academic biology laboratory to get picked up by the private sector, converted into a medicine, to complete all the rounds of clinical safety and efficacy trials. This is a key part of the investment in basic science into disease, is that we don't produce therapies that can be administered today. It's an investment, a long-term investment into novel solutions to these diseases. And so I will end here, and I'm very happy to take any questions you may have. But just to summarize, the major goal of our research is to discover novel targets for new cancer therapies that work by changing the identity of cancer cells from their one identity into potentially a normal cell fate. This is radically different than chemotherapy, radiation therapy, and surgery, I hope for obvious reasons. A large aspect of what we do is to study the details of how molecules work. We want to know how every one of these machines engages in the control of DNA function. And there are thousands of these molecules. We work on lots of different cancer types. This is no small task, but this is really our deep expertise that we apply to the study of these cancers. Now, we are applying this concept. We began in leukemia, trying to turn leukemia into normal white blood cells. We've been extending this into other malignancies. One that I'm very passionate about is called rhabdomyosarcoma. And this is maybe of interest to you all, because this is a cancer that I never intended to study. And it was really patients and families in the local Cold Spring Harbor, Huntington community who suffered and lost children to this disease, or even beyond that throughout Long Island, that really motivated us to start working on this cancer. And they told us the compelling and kind of terrible story of losing children to these cancers, and formed foundations to raise funding so we could really do something about it. And we've been really hard at work to apply what we've learned in leukemia to this disease. This is a cancer that is a cell that should have formed skeletal muscle. You may not have heard of this cancer because it mostly afflicts children and young adults. This is not a cancer that is more common with age. And we've been working towards trying to, and right now patients are treated with chemotherapy exclusively when the disease is metastatic. And obviously these are children. The chemotherapy is as harmful as the disease itself. So we've been working towards an idea of converting this cell, because it's a cell that should have become muscle, but it can't because of its genetic alterations. can we apply these principles to try to convert these cells to skeletal muscle? And we have reason to believe that if a cell becomes muscle, by gaining the ability to contract and become a terminal muscle cell, it will lose the ability to grow, metastasize, and spread. And we think it could be part of a way to cure this disease that we're heavily pursuing. Something we can talk more about, but we do this in the ivory tower of academia, and Cold Spring Harbor is in many ways the idyllic, bucolic environment to do discovery research, and I love that aspect about it. But I want to emphasize that this is clearly not just an academic thought experiment. We work hard to try to take these discoveries. Of course, we publish them, which is essentially like posting it on Facebook. It's available. Anyone can find it. A company, another lab. This is an open source model to research. That's what we love about it. But we also try to work with pharmaceutical companies and engage them strategically so that they can leverage our knowledge if it's useful to them. It can be. In ways where we can really integrate, work together, and not just we publish the papers and they use what we produce to produce medicines. And I think we're getting better at this, and I can say more about that. And then finally, Cold Spring Harbor is as much a discovery research institute as it is an educational institute. Training the next generation of biology researchers. We don't have undergraduate education. We have Ph.D., only Ph.D. education on our campus. Many of the scientists I've trained over the years now run their own research labs at other institutions throughout the world. There's a limit to how much I can accomplish in my own lab, but by training talented young people to perpetuate the science and the rigor and the ambition is really how this process continues. And naturally related to training is of course educating the public. And I would say this is important now more than ever, that those who are not scientists understand the benefits of investment in academic discovery research. So with that, I want to thank you all for being here and listening to me, and enjoy your evening. I'm very happy to answer any questions you may have. Thank you.